Okay, hello and welcome back to Ask an Archaeologist. I'm Sarah Kanza, the host of this afternoon's show. And Ask an Archaeologist is a series of live stream interviews co-hosted by the Archaeological Research Facility and the Phoebe A. Hearst Museum of Anthropology that we're putting together for Cal Week 2020, which is this week. Each day this week at 11 at 2, we're interviewing a UC Berkeley archaeologist and answering audience questions. So before we start the interview today, I wanted to let you know how you can ask questions. If you open a new browser window and go to sli.do, which is sli.do, and you can type in ask ARF, ASK-ARF, and click on the join button. And there you can type your questions and we will see them and ask them later on in the interview. And I'll remind you of this partway through the interview as well. So this afternoon, we're happy to have AJ White with us, who is going to talk with us about what we can learn from studying ancient poop. AJ is a third-year graduate student at UC Berkeley, and he uses fecal molecules to reconstruct ancient population changes. He's also interested in how climate change has impacted people in the past. He has worked in Egypt, Vietnam, the United States, Jordan, and he is currently working in his living room. So welcome, AJ. And we're delighted to have you. Thanks for joining us. So I'm sure the burning question on everyone's mind is, how did you get into this particular area of research?
Yeah, it wasn't something that from, day one, I was like dead set on it. I think in archaeology, you kind of have to keep an open mind for, what opportunities are out there. And when I started grad school at Long Beach State before coming to Berkeley for my master's in geology, my advisor there had said, hey, check out this cool paper that uses fecal molecules to reconstruct ancient population change. What do you think about it? Do you think this is something that, maybe we could do? And I thought about it and I was like, you know what, there's like a huge kind of emptiness in terms of like what we can do with poop in archaeology. And I mean, for a long time, people have looked at coprolites, which is, essentially fossilized poop or incredibly hardened poop. But there hasn't been a whole lot done on at the molecular level, and looking at what remains when everything breaks down except for a small amount of these incredibly persistent molecules. And I just thought, hey, that's like a huge kind of place that we could go and check it out. And we tried it out at a place called Cahokia in Illinois, and it worked really well. I've since then tried it out in Jordan. It's been a little not as well, but we can get into exactly why that is. And I think it's good to talk about, why some things work and don't. But yeah, it's something that kind of fell into my lap, and I'm glad it did, because I think there's a lot of things that we can do with poop.
I think that's fascinating because, when we think about archaeology, we think of all the more, the finds that are more harder, like bones and pots and that kind of thing. And we don't think of the molecular things that you could study. And that's much more recent research that's being done. Can you talk about how do you find these traces? Where do you find them? Do you have to find an ancient toilet? Do you have to go to a place where people were going to the bathroom, or where are these things everywhere? How does it work?
Well, yeah, so don't think it's super gross. I mean, the first thing that people think is that I'm actually going out there and just like scooping up some poop, just getting it. It's not like that. So we're talking about poop that has almost completely gone away. And the way to imagine this is that there was a time before toilets, right? And there's a time where people were mostly practicing what we'd call open defecation, which means just going out and doing their business like you're camping. And so when that happens, there's poop on the landscape out in nature. And after it rains or something like that, it'll break it up into little tiny pieces. And a lot of those tiny pieces will get eaten by microbes. But there are certain little tiny pieces of poop that microbes find incredibly unattractive. Just like we find all of poop unattractive, microbes find a little bit of it. These very complex molecules that are very rigid. In particular, they're the breakdown product of cholesterol. And as you all know, cholesterol is something that's hard for us to break down. It gets caught in our arteries and causes big problems. So the same idea, it's very difficult to break down. And so they just leave it. And so it's a molecule that and the specific molecule is called caprosenol. And it's something that, because the microbes don't want it, it just lasts in nature for a very long time. Now, it's very hard to kind of, if you were to just go out to an archaeological site, it would be and this is an area where, before toilets, which until, that was actually much of human history until like past couple hundred years, really. It would be hard to just guess where people went to the bathroom, right? You'd be very lucky if you just put your shovel in and that's where you found these molecules. So to get around that, we target areas where these might have collected after being washed away by rain. And so if you're in somewhere where there's like a lake nearby, that's a catchment. That's an area where stuff naturally just gets deposited. And so we target lakes, wetlands, low places where the idea is maybe only a small fraction of what was originally in, one stool will make it in, but it's still something. And, we go and we, collect big sediment cores, which are, columns of mud. And then you can look at how much of these molecules are present at any depth. And the idea is, hey, if we have more at position X and it's more than position Y, that probably means there are more people here. And if you're lucky, you can tie that into maybe there's a migration story in the area, and they're like, oh, that's when it happened. That's when people came here. Or maybe it's the opposite. Maybe you can see where people leave. So yeah, that's the idea. It's not actual poop. It's very specific molecules that might accumulate in specific environments.
Great. And so I know you had a recent paper that came out in what, antiquity, American Antiquity?
That's correct, yeah.
Yes, that about your work at Cahokia on this. Could you explain a little bit about what that looks like?
Sure thing, yeah. So we first went out and went to Cahokia. And by the way, Cahokia, for those who aren't familiar with it, is like one of the biggest archaeological sites in this country. It's home to dozens of these monumental mounds. And to make dozens of monumental mounds, you need a lot of people. So it's thought that this area was a city that, maybe was home to several tens of thousands of people. And what we did is we went and right outside Cahokia, there's a lake, and we collected sediment from that lake and did the process that I just explained, identifying where these fecal molecules are in the mud, and then trying to link that back to the population at Cahokia. And I did what I think 99% of most archaeology in the area does, which is I immediately went to what's called the Mississippian time period in terms of where my brain went. And that's the time period where most of those mounds were constructed, when most of the people were there. There's a whole lot to say about it. And there's really great research. If you're interested on the Mississippians, you're gonna have a lot of fun learning about this stuff. It's totally fascinating. But the thing is that there was stuff that happened after the Mississippian time period that very few people have written about. We know for sure that there was an indigenous presence in the area at the time of European contact, because the French missionaries established a mission at Cahokia, like actually on the site. And there's almost no archaeological presence of this, very limited. And there's no archaeological evidence that's been put forward to where the people that were living who were to be associated with this mission. And it must have been enough people to justify going all the way down there and building a mission. So we know there's a substantial amount of indigenous people around this area, but there's very little research about it. So what we did is we said, hey, we don't have archaeology, but we do have this fecal stable population reconstruction, and it doesn't just disappear at 1400 AD. There's 300 years of additional demographic changes. It looks like we do hit a low period after the Mississippian occupation, which is what everyone talks about. But starting around 1500 AD, the population numbers from our data seem to go up modestly, not a whole lot, but they go. And the idea is maybe that's when we start to see the movement of this population that was present at the time of European contact, which we think is probably a group of the Illinois Confederation, who were called the Cahokia, and that's how Cahokia got its name. And so we're able to kind of tie that into this whole very under-discussed aspect of the local history. I mean, it's generated, it's one of those things where, like, how much can you say without archaeology? Mm-hmm. And I'd like to say that you can. There is a story to be had, even if we don't have stuff that our eyes can see in the ground. There's stuff in the ground that we can't see that still leaves a record. So that was kind of the fun thing about it, you know.
And can you explain why there isn't archaeology during this time when there were actually people there?
Yeah, I've tried to. It depends on how much you believe me. What my interpretation of this is that the two different indigenous groups that were present in this area at different times, you know, the Mississippians under, you know, the traditional period of Cahokia, and then those came after, which are likely the Coquia, which is a member of the Illinois Confederation. It's very confusing. I'm sorry for that. But they had very different lifestyles. And so you know, during the Mississippian period, this was people who were extremely sedentary, so sedentary that they were building, you know, giant mounds that were as big as the Great Pyramid of Giza. In terms of their base, at least, not their height, but the point is it's big. And you compare that to the Illinois peoples, and they were mobile. You know, they were semi-sedentary. So they'd go on these long bison hunts in the summertime, where the you know, the groups would get together, and it'd be this big outing. And the point is, they're not where they are for the rest of the year. And so maybe there are big camps that are being built in the fall and the spring. In the winter, you know, resources are more scarce. People would go off to live in small family groups. When you're in the winter in a small family group, you know, it's just, you know, maybe 8 people, I'm going to guess, you know, a small number of individuals. You're not producing as much archaeology. You know, the less people that are in any sort of spot, you're not going to be leaving as much stuff behind. It's going to be a lot harder to see. And so Mississippian stuff is obvious. It's a giant mound. You know, you're going to find it. Stuff from the later time period might be a little more subtle. And I think it either is stuff that hasn't been found, it's stuff that's been destroyed by a fair amount of post 1900 just expanse. This is right outside the city of St. Louis. So there's been a lot of just highway building, housing development, stuff like that. And finally, I wonder if some stuff that might be from this time period has been dated Mississippian, just because if you don't have diagnostic artifacts and you find like a small scatter of chipped stone, you might think, oh, well, everything else is Mississippi, and I'm going to check the box for Mississippi and go on with your survey. I mean, that's super. That's just speculative. But I wonder if there might be a little bit of this kind of little recording bias going on. So yeah, I mean, I could go on with my theories, but I hope that gets the idea across.
Yeah. Well, let's pause for a sec to remind listeners on YouTube that you can ask questions by going to Slido, SLI.do, and typing in Ask ARF. And you can pose your questions there, and we'll see if AJ can answer them. But in the meantime, can you talk a little bit about how animals, do they produce this too? And can their poop get in the mix and mess things up?
Absolutely. So what I just told you about, you know, this whole method is relatively new in terms of using these molecules for population estimates. And there's always a point where, you know, the method's looking great, and then everyone kind of realizes, okay, here are the imperfections. Here are the places where it kind of has a fair amount of limitations. One of them is what you just mentioned, is that humans aren't the only ones that make these molecules. Now, for everything that I've seen in the literature, humans make a way more caprosenol than anything else, on the average of, you know, 10 times as much. But there are certain animals, they tend to be large domesticated animals that we've tested, things like sheep and pigs. Cows make a little bit too. Will produce a little bit. And so the point is, how can you tell whether you're dealing with a city of cows in the past or a city of people? And there's certain kind of ways around this. And at Cahokia, we were able to kind of make the argument that, okay, this is before European contact. There's no large draft animals in the area. The only domesticated animal is probably going to be a dog. And dogs, we know, make almost, they don't make any, actually. So carnivores don't really seem to be making this molecule. So we could say humans are the big animal on this landscape. And, you know, whatever is being produced, there might be a bear that goes by occasionally that makes a little bit of it, but we can be pretty sure that this, whatever controls the changes is people. Now, if you're in an area where there's a lot more animals that are present, that are, you know, particularly domesticated animals, like those I just mentioned, then it becomes a lot harder to pull apart the human and the animal signature. There are ways of doing this in terms of looking at more molecules, particularly bile acids, and looking at various ratios of these different molecules to each other to say, if you have this number here and this number there, it's probably a pig, you know, and so there's ways to get more specific, but you can't completely be 100% certain that, you know, everything you're seeing is human. And we ran into this problem a little bit on more recent work that I'm doing with my advisor, Lisa Maher here at Berkeley. And we tried this out at her site, which is in Jordan. And it's very old. It's about 20,000 years old. It's an amazing archaeological site. It seems like there's a lot of people there. However, since there's been a lot of people there 20,000 years ago, there's also been a lot of camels there that have just been going across the desert. And so we were wondering, does camel poop contain some of these molecules too, and could that contribute to what we're seeing in the sediment? So we tested camel poop, and it turns out, like cows, they make a little bit too. So, you know, what we think is happening is that over time, their poop is broken up and kind of percolated downward into archaeological sediment, and it makes it really hard to distinguish, you know, what's going on with people and animals. It's a real mess. And so what I hope to do now is kind of go out and say, okay, here are some of the limitations. Here are some of the places where I think it works really well. I think if you're like on an island, somewhere where you know exactly what's coming in and out. I think if you're in the Americas, in a pre-contact setting, it might be a pretty good way to do. But in the old world, it's a little more complicated. So yeah, that's what we're getting at. That it's there's a lot of limitations to be discovered, and we're kind of getting into that.
Mm-hmm. And so how old is this approach? I mean, like, what does your research community look like? What's the size of, do you go to conferences that for people who just do this, or is this a really emergent?
It's just the men's restroom, you know, that any conference you go on, no, anyway, but so what we do is, where this all started was in the 1960s or so, people started using caprostanol for sewage contamination studies. And they still do this today. You know, like if a pipe breaks near a port or something, or somewhere where people go swimming, they'll go out and test the water. And if they find caprostanol, it's like, okay, no one goes swimming in this water. There's been a sewage leak. And so that's where it started. And then archaeologists beginning around the 90s realized that this molecule doesn't just indicate modern sewage and modern human waste, but also is a molecule that can persist for a long time. And so they started first using it as like an indicator of a human presence. And so particularly for pooping. So, you know, people used, okay, let's say you've got an ancient fortress and you want to know where people went to the bathroom. If you find an area where you just find a ton of these molecules, then you've found your bathroom. And so it's stuff like that. So there's, you know, studies from Roman forts that were finding where the privies were. It was used to try to find out where, you know, fields were manured and other fields were not. But in terms of using it as a population indicator, that didn't really start until 2012. As far as I know, that's the first paper that went out and said, hey, we can use these as a way of tallying how many people were here in a kind of relative sense. So in terms of population, it's less than a decade. But the whole thing has been around, and there's a kind of cool history to it. Yeah, and the fact that it still is used for finding sewage pollution, it still goes on, just as it has been for 50 years.
Cool. So when you talk about like finding it in different parts of a site, say, I know this isn't necessarily the research you do, but if we were at a Roman fort or something, what kinds of tools would you use to sample areas? I mean, what kind of controls need to happen during the excavation to allow you to do your work?
Yeah. I always joke that it's good to wash your hands, just because, yeah, you never know what sort of contaminants you might be bringing. You know, we're dirty people, as this whole, you know, outbreak has shown us. You know, you can never be too sure about what you're carrying with you. But yeah, the first thing is, yeah, you don't want to contaminate anything. You don't want to be eating anything that has cholesterol and, you know, be touching anything with that. So if you just had like a pepperoni pizza, not a good idea to go out and do your sampling. But let's say that, you know, you got, I wear gloves on when I'm in the field, and if you're not doing it the approach we talked about before, which is you go to a lake, right? And so if you're in an archaeological site, it's the way we've done it is much like any sort of excavation where you have your profile, your side wall or what have you, and you can pick depths that you want to sample. And this isn't anything you can see, right? So you just need sediment. Typically we fill up, at least for what I do, is fill up one of these is about, maybe 15 milliliters or a volume of sediment. And from that, we don't need a whole lot because we're going to take what the sediment is and we're going to go take it back to the lab. We do what's called a solid to liquid extraction. And so you take the dirt that was in here and you want to get it into a solvent that will strip out the molecules you're interested in. And now it's out of it's out of the mud and it's into something liquid that machines can deal with. And then you do several controlled chemical reactions to further isolate the molecules you want. And then you put that in mass spectrometer, which is an extremely sensitive instrument that can tell you how much of these molecules are present to the nanogram. So you don't need a whole lot. And in terms of the sampling procedure, it's pretty simple. The analytical procedure is a little more involved. That's the part that takes the time.
I see. And so it sounds like carnivores don't produce any trace of this, but herbivores and omnivores do. And so is it refined enough to be able to tell, like, if there's a human population that is eating more of one type of food or more meat or more plant material, like, can you actually distinguish that from the amount?
No, so there's I'm going to break your question into two parts. The first one is that I don't think we have enough data yet to make and I kind of, this is my fault because earlier I said all carnivores don't do this. The problem is we've only sampled, as far as I can tell, perhaps under 50 animals specifically for this that I'm aware of. A lot of them are ones that we can easily get our hand on. So Steph, if you go to a farm, just sample everything there. So there needs to be, I think, just a lot more survey of just, what the different of the animal kingdom to find out, okay, well, is it true that we can say carnivores in general, or am I just basing that off of a dog, which in that case I was. So I think we need to expand the amount of animals that we're studying, but then let's say that it does pan out where there are differences based on these kind of diets, right? Yeah, it might be possible to look into that. And I think it would be difficult because it would be a trade-off between, let's say, you had someone who is ingesting very little cholesterol. So conceivably, they would produce less coprostanol, right? The byproduct of the cholesterol breakdown in our guts. However, that might look, if you had 100 of that group of people, would that just look like 50 people who were eating, more cholesterol, right? Because it's not like there are multiple variables here. And so it would be difficult to pull apart whether it means more people or a different diet. At the moment, the assumption I make when I do these studies is that people have a similar diet through time. Of course, there's going to be differences in there, but you have to kind of choose some sort of assumption in order to make interpretations. Otherwise, you're just going to say, there's too many variables and leave it there. But one thing I've joked about doing, and I actually might want to seriously do this, is actually try that out myself and have like these different diets and then just test what comes out to see if there are noticeable differences. Because one thing that's good to keep in mind is that we produce cholesterol as well. So no matter how vegan you are, there's still cholesterol in your body. Otherwise, you'd be just a bunch of jello because you need cholesterol in your cells to have structure. So we make cholesterol anyway. So it's complicated. I'm not sure if that completely answers it.
No, it's fascinating to hear about this new area of research. And I love the idea of being able to see beyond what's actually in the ground, what's being excavated, and to be able to do these types of analyses. A listener has asked, what's your favorite project so far working in this field?
Gotcha. Well, I've been very fortunate to do these fecal stanol analyses in multiple places. In Vietnam, we did a really cool project on an island and looked at sort of the the timing of when the greatest populations were there. At Jordan, we've learned a lot in terms of the method, but I'd say that in terms of what has, I'd say, produced to me the most interesting findings is from Cahokia, I was talking about earlier. And one thing we haven't talked about is that, from the Cahokia study, we were able to say, okay, in our sediment core, here's what's happening with the fecal molecules. But we're also going to say, okay, well, what's happening in the environment based on what we have in this lake mud? Because, we can see things that were going on in the environment just by looking at the mud with our eyes. There's like a very obvious flood, and it looks like this real gray pulse of silt that just shows up. And right after that, our fecal molecules start to be in decline. Now, in addition to that, we sampled the mud for oxygen isotope analyses, which is a way of getting what rainfall was like in the past. And we also find that around the timing of that flood, it seems like rain started to change and it became drier during the growing season in the summer, which would be a big problem if you're making, if crops are what's feeding your people. And so it seems that climate change was happening, and it was having some effect on people living in this area, in addition to what was happening, in their society, and completely cultural realms as well. So what I really love about that is that I think we've been able to make a really clear sort of argument for how the environment and people go together through this method. And I think it can be quite powerful. And I hope to encourage people to try this method out for, among other reasons, just that you can really connect, the climate and people in a way that it's hard to do otherwise. So I've really enjoyed, making those interpretations and being able to pretty confident about them.
Well, that leads nicely to another question about what, if people on archaeological projects should they all be doing this? Should they all be collecting these samples, even if they don't have someone like you on their team? And how would they go about that? Is that something they could do?
Yeah, so what I feel like I'm learning now is that this isn't necessarily a method to just be applied universally to, every single archaeological site. Due to the way that this molecule preserves and due to how other animals can affect its interpretation. So I think that if you're an archaeological site and you've got a nice wetland nearby, that's an area that, go get a core of it. And sometimes that requires specialized equipment and training to do. But if you have a nice sedimentary archive right by your site, that's going to be an area where you can be pretty confident that the stratigraphy is real intact. There hasn't been a lot of disturbance. Things are going to be in there. The problem with an archaeological site is that it's porous sediment a lot of the time, right? If you're dealing with sands or silts, even stuff where stuff can mix around. What I think was happening at our site in Jordan is that modern or near modern molecules are percolating into archaeological sediments, which makes it a real problem. So if you have somewhere where it's nice and tight in like a waterlogged area, I'd recommend it. And you also have to consider the archaeological context as well. Are there a lot of animals that might produce this in the area? Are you in Africa, Europe, or Asia? Or are you in somewhere where there might not have been as many large animals in that area? That would perhaps be a better option. So I'd say, yeah, think about the context. And if it checks those boxes, then yeah, and you can, there's plenty of people who are starting to do this when you can, collect the samples and, give someone an email who's published on this and say, would you be interested in analyzing this? And it is an expensive method, I might say. So per sample, it can be pretty pricey. It's not something that you can just say, oh, well, we'll just throw it over there. It does take some money. So it's something.
And does the sample have to be sent to a lab and processed by someone like you, or do you send it out somewhere to be analyzed?
Yeah, it depends on how comfortable you're with chemistry. I'm not a chemist. I'll go ahead and just say that. So to me, what I'm doing in the lab feels sophisticated. To a real chemist, they're like, oh, that's undergrad chemistry. That's not a big deal. If you know your way around a lab, you can perhaps do the extraction procedure I imagine that any sort of university campus will have some sort of way to have this solid to liquid lipid extract, which is what you're doing. That's what you're making. Now, once you get to that part, you need to run it on a gas chromatography mass spectrometer. That's something that, I don't do that part because that requires a fair amount of training and understanding of a very delicate and expensive piece of equipment.
Mm-hmm.
So I'd say that at that point, you almost certainly need to send it out to a lab, but you might be able to do the extraction point and then send them the sort of the final project before it goes in. As I'm saying this, though, I think it is best to perhaps send it out to someone who specializes in this, even though theoretically
Right.
you could take care of it.
Good. And so along those same lines, then in doing this work, do you look at actual poop? Is another question that came in.
I have
guess
seen it.
What's that?
I've seen it before.
Yeah.
No. So I do not look at actual poop, and that's something that I get asked a lot. There's a whole tradition of looking at coprolites in archaeology that is much older than what we're talking about here. And there's a lot of great things that can come from looking at actual poop, looking at the stools in terms of looking at diet and looking at macro remains inside of it, as well as looking at parasites and looking at how people's health was like in the past. But what we're talking about here is all at the micro level. This is all stuff where it just looks like dirt. And that's what's kind of fun about it too, is it's like in any sort of volume of dirt, there's all sorts of stuff in it. I mean, it is crazy dirty. That's another problem with running this on the mass spec, because there's just so much stuff in dirt, and it has so many sources, and it's totally its own little world in there, and it blows my mind.
Yeah. Well, it's so great to hear about this from you, and it's fun to learn about something totally new. And I'd like to thank you so much for joining us today for this session of Ask an Archaeologist. So thank you for coming, AJ. It was fascinating. And thank you to the listeners, the people who joined us online. I'd like to invite you all to our next Ask an Archaeologist, which will be tomorrow, Tuesday at 11 a.m. And it's going to be with Scott Byram, who will be talking about how archaeologists use ground penetrating radar to map buried artifacts and ruins before they begin an excavation. So another chat about archaeology without actually looking at artifacts. So I hope you're able to join us tomorrow. And again, thanks so much, AJ, for joining us. It was fascinating.
Thank you.
Okay.
All right.
Bye. Oh, nice. Oops.
All right, goodbye.
Bye, thanks, AJ. I don't know, I guess she turned it off. Whoop.